back everyone to the online conference, Food Innopolis International Symposium 2020. And now before we start the panel discussion, if there is any question, please send it to Q&A box. The speaker will be pleased to answer all your questions at the end of the session under the topic, Advanced Microbiological Control for a Reinforced Food Safety.
Now, please welcome Ms. Amparo de Benito, research scientist, AI and IA's man. Please welcome.
Okay. First of all, I would like to thank the organizers for inviting me to this symposium. And as they have said, I will be talking about the advanced microbiological control for reinforced food safety. So first of all, I would like to tell you about the place where I come from. I'm working in a research center that is called AINIA and is located in Spain, in Valencia, that is located in the East Coast. We are an association of 741 members of food industry mainly, with 27 governing members. The areas that we work, they're very broad, and the sectors as well. We work in fields such as biotechnology, food analysis, communication technologies, consumer science, environmental and energetic technologies, packaging technologies, automatization and robotics, extraction technologies, consultancy and training. And in the sectors of food, food packaging, chemistry, energy, cosmetics, and pharma. We are a very large place in Valencia. We have 13,000 square meters of laboratories and pilot plants. The laboratories work in most of the analysis that they are in the European legislation for food criteria, and we are accredited. And the pilot plants are very broad. We have lots of areas such as biogas, bioproduction, hygiene, extraction by supercritical fluids, artificial vision, culture of microalgae, and, for example, microencapsulation. So there is a broad range of different research work that we do. And to talk about the topic of the talk, I would like to first of all give you a view about the new emerging microbiological risk related to food production. Changes in the food production, consumption, and environment have contributed to the changing nature of the food hazards we face. Some of the most significant changes include large-scale production, changes in habits, and increase in the consumption of raw foods. This is especially in the European and in the US market. Increasing importance of international trade, more food consumed outside the home, and preference for minimally processed food, as well as increasing susceptibility of the population, as especially in Europe, the population is aging, so they get more susceptible to illness. And there is a big worry about the antimicrobial resistant microorganisms that is also threaten the safety. And of course, also is influenced by the climate change. So in this situation, food industries faced future problems, and they have to be prepared with advanced methods to control the microbiological safety. So why is necessary alternative rapid methods in food microbiology? Well, food industry needs to release safe products in as little time as possible, because there is a product demand which is always increasing. Many products are self-life, so especially with the more consumption of fresh foods, and so you need fast results to put the products in the market. Also, there is a cost associated to the storage, so it's important to release the food without having to wait for the results. Also, it's important to give solution to problems with fast decision making. It's also necessary fast diagnosis for control plants. They have to be very agile. And also it's important to have fast answers to a food crisis. So the techniques have to be more accurate and more versatile. When we consider the introduction of fast alternative methods in a laboratory, you have to be aware that there are advantages and disadvantages. For example, the advantages of fast alternative methods are the speed, obviously. Also, there are automatic methods and they have high throughput. Also, they are really reliable in their interpretation, easily to interpret and reduce human error. But on the other side, the traditional methods are also have some advantages. The reference methods for criteria, for example, in the European legislation, we have reference methods ISO and CEN, which are based on traditional methods. They're also universal because ISO produce methods that they can be used in the whole food chain. And that means that they try to have as little limitations as possible for their application. There is no an initial investment in a big machine, expensive machine, which will measure some activity, because it relies on the growth of bacteria in culture media. So in a way, they are inexpensive, but I put a question mark here because in the long run, because it needs lots of labor, in the long run, fast methods could be cheaper. The drawbacks of the fast methods are obviously the initial investment, more infrastructure, and the expense of the reagents, although there is, with time, this is getting better. And also there's some limitations in the application because some food matrices are not compatible with the way it's measured, the technique. Also, there is initial rejection sometimes when you change from using the traditional method to something fast. And this is getting less and less, but in some cases it's difficult to convince, for example, your client to release your products with a fast method. And of course, the traditional methods, the drawbacks that they have is that the results are between 3 and 5 days. They need more handling by the laboratory workers, and that also is important because it's possible interpretation errors because it relies on people. So one thing important, once we have said this, is that fast analytical methods should be validated against a reference method. And if it's a commercial method, it's recommended to be certified by a third party in accordance to a recognized protocol. There is a protocol for alternative method validation, that is the ISO 16140 Part 2. And it's important as well to get a certification by recognized companies such as AFNOR, AOAC, there are some others, because that will make them valid and equivalent to the reference method and also more accepted. So when we are talking about alternative rapid methods, we have two possible options. One is the method that is shorten the steps within the conventional method or is based on conventional methods, or is using a completely different methodology. So in the first case, we have chromogenic or ready prepare media, faster protocols, miniaturized methods or automatized methods, immunomagnetic separation or confirmation kits to identify the bacteria once it's detected on the plate. And in the case of methods that are used in different technologies, it can be based on immunological method, genetic methods, flow cytometry methods, or even mass spectrophotometry methods. I will introduce here a few things about hyperspectral imaging, which is something that is not already in the market, but that there are some research going on in order to be able to detect microbiological quality online. So when we are talking about alternative rapid methods that shorten the steps, we can have the dehydration media. In this case, you get a media that is ready to use, and time is shortened because the colonies can, in some cases, that you don't need to confirm colonies, so there are faster methods and they can be automatized reading. Then you could have immunomagnetic beads in order to save the enrichment step, which you will save 1 day from your protocol, because it captures the target microorganism by antibodies. And these beads, because they're magnetic, then they can be taken by a magnet from the broth. And another system is miniaturized systems for enumeration and confirmation. So you can identify your target bacteria, or you can even do the whole analysis in a miniaturized system, which it will save time because the cultures are so small that the bacteria grow very quickly. When we're thinking in alternative rapid methods based in other technologies, we have, like, for example, immunological techniques. In this case, they are based on antigen-antibody reaction, so they are very highly specific. Normally, a second label antibody is used, and the detection is after an enrichment phase of 24 hours. This is important because these techniques are used for detection of bacteria such as Salmonella or such as E. coli or Listeria, which are, you need to ensure that your food is free from this bacteria with a high sensitivity. So you will say that they are not present in, for example, 25 grams. And for that, you need an enrichment because these techniques cannot work directly with the food. You need an enrichment in order to bring the numbers of the bacteria higher in order to detect them. And in this case, many protocols, they recommend confirmation of positive results. So it's used for an screening more than a total result. They are easy to operate. There are many machines based on immunological techniques. They're normally automatic reading, and also they are in continuous development because of the recognition site could be other things apart from antibody. There could be bacteriophages, etc. Then another alternative rapid method that is different from a conventional method is the ones using genetic techniques, that they are very, very popular now. For example, PCR real-time or quantitative Pcr. They are based on the specific detection of DNA or RNA after reverse transcription. Is high specific and sensitive. Also, because it normally is used for bacteria that they are in pathogen bacteria, and you need a detection in 24 grams, the detection will be done after an enrichment phase of 24 hours. They may need the confirmation of positive results because the conventional PCR real-time detects non-viable cells. So that cells could also give you a positive and can be used for quantification, and also allows you to do your own development. For example, we have this system in the laboratory where I'm working, and we have done our own detection assays for example, spoilage bacteria, etc. So they are open for your own research more than other systems. An alternative of this PCR real-time is a PCR that detects viable cells. In this case, will only detect and amplify DNA from viable cells because it's based in the use of DNA intercalling agents such as PAM or some other reagents. And it saves the time of confirmation of positive results and avoids false alarms. So the way it works is that you will get the mixture of cells that they are alive and dead, and then with this agent, there will be a binding to the DNA, and this binding will stop them from amplifying. So that's how it works. And there is another option as well for PCR, which is digital PCR. In this case, the structured DNA from the sample is distributed in a microchip in order to separate many different aliquots that will hold very little amount of DNA. In some cases, you won't have the target DNA in some of the wells. So this will give you a pattern of positive and negative reactions, and this will help you to quantify how much of the target you have in your sample without standards. So it works like the most probable number, but in this case with PCR reaction. So it's very powerful. It has high precision than the conventional real-time PCR, and is tolerant to inhibitors. And now I'm sure that you have heard as well about the new generation sequencing. These techniques, they are very powerful to sequence the genome of whole bacteria, or also you can sequence a mixture of bacteria. So when it's applied to food safety, it will give you a rapid study of the microbial population of the sample. The detection of pathogens and spoiled microorganisms can be done without a previous PCR technique for the target bacteria. So it's a very good way to find what is the cause of the problem without a previous knowing what are you looking for. So it's very good for epidemiological studies. The results are obtained in real time because in a few minutes of running the sample, you start seeing some of the results. But you have to run it for a long time to get the whole population identified. It gives you a relative quantification of the microbial population, so it's very good also to apply in fermentation processes, for example, because it will give you a percentage of different bacteria and telling you what is the mixture. And of course, there is a possibility of genetically characterizing microorganisms. So there is an important application as well for bacteria that they are used for a purpose. So here is what you will normally obtain with these techniques. This is an example of one of the essays that we did at work. So they are quite powerful techniques. Another technology that is used for identifying fast identification of microorganisms using a different technique is the flow cytometry. The flow cytometry detects and quantifies the cells of a sample one by one. You will have a flow and a stream that will pass the solution with your bacteria through a channel, and this channel will be there will be a laser hitting this pass-through channel, and this will measure optical properties. So the light emitted by fluorochromes and light scattered by the particles in the flow. This is quite good because it will differentiate cells by size and complexity, and it will give you an automatic reading. Despite the fact that it seems very, very complex, there are already systems like this in the market. And also there is another system that is the alternative rapid methods based on mass spectrophotometry. In this case, the equipment I would like to tell you about is MALDI-TOF, matrix-assisted laser distortion ionization time of flight. So this is mass spectrophotometry that allows protein screening directly from intact bacteria. The reading will be automatic, and it will give you a rapid identification of microorganisms. Here in the picture below, I show you the equipment that we have in AINIA. It's a very useful tool because within minutes will give you the identification of your bacteria when you have it isolated from your plate. So it requires very simple preparation on a support, and it will give you a very fast identification of your target bacteria. So it's very useful for confirmation, but also had applications to do detection directly from your samples. For example, it has been applied to identify microorganisms from urine samples that when you are only expecting one bacteria. And finally, the hyperspectral imaging techniques. This is still under development. Is based in the non-destructive tool for quantified microorganisms directly from food surfaces. So it's based in a multivariate statistical model for predicting microbial contamination of the food. It can allow real-time determinations. And this, the idea is to use this technology the same way as you already using similar technology for foreign body detection. So by passing through the sample, the image will capture the signal in order to be able to discriminate the ones that they have more load of bacteria in comparison with others. Of course, this is still under development. There is no a machine already in the market to do this, but this probably is one of the future developments because we need more and more faster methods to detect microbiological problems in foods. And that will allow us to do it on real time, and also with a non-destructive tool, you will be able to pass through all the samples and discard the ones that they are not complying. And AINIA is at the moment looking at this technology and trying to get some funding to develop this kind of technology. So thank you very much for your attention, and if you have any questions, I will be happy to answer them.
Thank you very much, Ms. Benito from AINIA, for a very interesting information.